Hi, everyone, and welcome to this short interview with Anthony Lodge. So Anthony was a key contributor to the EU regulatory affairs strategy for Kite Pharma's CAR-T products, Yescarta and Tecartus. And he also had experience with stem cell-based product, having worked at GSK and Casey. He's now Senior Director, Regulatory Affairs at T-Knife Therapeutics. So Anthony, welcome.
Good
So
morning,
Anthony,
Fabio. Thank you for giving me the opportunity to speak to you this morning.
it's a pleasure. So I was really looking forward to our interview. So I'd like to make this as useful as possible to a broad audience. So can you start by telling us what's the role of regulatory affairs in drug development?
Yeah, thank you, Fabio. Regulatory affairs is actually quite a broad discipline. The essential role of regulatory affairs is to be the interface between a biotechnology company and the regulatory agencies who will approve the drugs that the biotechnology company is developing for use in either clinical trials or for marketing application. So I think that that's the key role of regulatory affairs, but within that, there's, various different types of work that we do to make that happen. There's two sides as well, really, to regulatory affairs. There's the clinical side, so that's the side of regulatory affairs who looks after the clinical trials initially. Also, typically looks after some of the applications to get regulatory designations that can support development programs such as orphan drug designations, such as prime designations, such as breakthrough therapy designations. And then, the clinical regulatory team will also support the clinical development team to put together clinical protocols that the regulatory agencies can approve to ensure that a clinical trial will go forward. The other part of regulatory affairs is CMC regulatory affairs, and that's the role that I do. So CMC regulatory affairs is all about supporting the manufacturing of a medicinal product. So within a biotechnology company, there's usually an organization called technical operations. The name differs, between companies, but technical operations is a good general term, I think. So technical operations encompasses everything that's focused on developing a manufacturing process, quality control procedures, etc., for a medicinal product. So you've got the process development people, you've got the analytical development people, you've got the quality control people, you've got supply chain, you've got manufacturing. So my role is to work with technical operations to develop a CMC strategy that will fulfill regulatory requirements that we can present in a technical dossier, so module 3 or an IMPD that goes to the regulators for them to review how the product is made, and for us to demonstrate to the agencies that the process is well controlled and is delivering a product of suitable quality that's also safe and efficacious to be used in patients in a clinical trial and ultimately in commercial use through marketing applications. So there's a lot involved in putting together that CMC strategy that aligns with the regulatory requirements presented in the regulatory dossier. Discussing the plan with the agencies through scientific advice type procedures, for example. So really, they're the two key disciplines of regulatory affairs. CMC regulatory affairs being my particular role.
Thank you. So now let's imagine that you're talking to a scientist who would like to set up a company to turn their research into new medicinal products, new cell therapy products. and they are trying to set up like to develop a regulatory affairs strategy, like a development strategy to IND, and possibly with an interested investor. And what would be your key three tips to set them to the right path from a regulatory point of view? Because I understand like setting up a company is a very complex task, but from a regulatory point of view, what would be your three key tips?
Okay. There are probably more than three,
I see.
but let me choose three for now. So I think the first one would be to hire people who've got, really good experience in the industry and in regulatory affairs. I think, to put together a good regulatory strategy and effective regulatory submissions, then it's really important to have experienced people to do that. I think for small startup companies, they've got the opportunity to use consultants. There's lots of consultants out there who are probably a good first start while the company is getting set up. But I think it's really key to hire people, either as consultants or, as full-time employees who into high-level positions who've got good experience that can execute on a regulatory strategy. That was number one. Number two, I think it's important to understand regulatory frameworks. Now, that is obviously mainly the role of the regulatory affairs professionals, supporting a development program, but I think also for an organization as well, more broadly, it's good to understand regulatory frameworks in US and Europe in particular. Not to the level, I guess, of the legal framework and directives and regulations, but what the agencies do is they develop guidance documents that delineate the expectations of what a development program should incorporate. So I think it's really important to understand the regulatory frameworks, be aware of the guidelines out there and use them to inform the strategy for your particular development program. So my third tip, and building on that knowledge of the regulatory frameworks, would be to actually interact with the regulatory agencies. So the European agencies through scientific advice meetings, or the FDA through meetings such as pre-IND or interact meetings. I think it's really important to talk to the regulators, certainly in early stages of development, pre-phase one, for example, to ensure that the way that a development program, the strategy for a development program is designed, is going to fulfill the regulatory expectations. So obviously there's the knowledge that comes from understanding the regulatory framework and the guidelines, but actually seeking that direct feedback from the agency is really important. So engaging in scientific advice is a key tip.
Okay, thank you. So then, like we often talk about potency assays and comparabilities, and it seems that they are the biggest hurdles in ATMP development. But I would like to ask you, what do you think are the most difficult, like the biggest challenges when it comes to CMC regulatory affairs for ATMPs in particular?
I think there are many CMC regulatory challenges with ATMPs. They're probably product specific as well, but comparability studies and potency assays are very important things to talk about. Let's think about why they are needed. So potency assays are needed to demonstrate that an ATMP will have biological activity, and we can therefore predict that it will be active in patients once they are treated with it. So a potency assay is a key aspect of the release testing for a product. But it takes time, it takes work to actually develop potency assays. Certainly in the beginning of a development program, there will probably be multiple potential potency assays that are under development until one can be selected based on being the most robust readout, but also correlating with efficacy in patients. And it's not until patients have been treated and until you can actually correlate that release test, that the data from a potency assay with the clinical data. So that's the challenge in early development. But certainly for a lot of the T-cell therapies, CAR T-cell therapies now, then interferon gamma expression is commonly used as a potency assay. And even if it's not perfect at phase 1, then at least something can be implemented. And I think it's important to be collecting data on biological activity, even if it's not assigned as a potency assay, from phase 1. So, implement potency assays or potential potency assays as early as possible, because it's important to correlate that activity with the clinical response so you can understand how the product is working. But you also asked about comparability studies. And comparability studies are important because manufacturing processes for ATMPs in particular are most likely to evolve during the development program. So most organizations will want to implement a few changes, improvements to the process. Maybe they want to change the way in which the cells are enriched from the starting material. They may want to change the actual culture conditions or use different materials, for example, just make various improvements just to improve the process, which is, a natural evolution of process development. So comparability studies are really important to show that when those changes are made, after patients have been treated with a product using one version of the process, that the quality of the product is not changed, certainly not impacted negatively from those changes. So that's the point of doing comparability studies, so that you can compare different flavors of the product from different versions of the process that have been used in patients during the various phases of the clinical studies. So potency assays are a key part of that, because it allows you to demonstrate that the product continues to exhibit biological activity. So the two things go together. I think because changes are going to be made throughout development, you need a robust analytical toolkit, shall we call it, to be able to assess how those changes impact the product, and potency is a key aspect of that. So the two things definitely go together. And I think another thing that many startup organizations probably need to be aware of from comparability studies is that the design of those studies, can be quite complex and needs to be set up appropriately to be able to generate data that can meaningfully conclude on the impact of particular changes. And that's, where it's important, going back to what we were talking about earlier, talking to the regulatory agencies, seeking feedback in scientific advice meetings on the design of comparability studies, for example, to make sure that the design is correct, the statistical approach to analysing the data correct, but also the analytical toolkit is correct to be able to generate meaningful data on process changes that are being made.
Yeah, because we need to generate the right data, not just like quality is more important than just quantity, I guess. So you would agree that potency and comparability are, let's say, among the biggest challenge, the biggest hurdles when it comes to CMC regulatory strategy for ATMPs?
Yeah, absolutely. Simply because, you need to be able to show the impact of changes on your manufacturing process, on the quality of the product, and the quality of the product is intricately linked to its potency.
Excellent. Well, thank you. And now I'm thinking back to what we were saying at the beginning, that you work both on like pre-approval and post-approval, but you work with the T-cell based products, the Yescarta, Tecartus, but also with stem cell based products like Streamvelis, at GSK, but also Holoclar at Chiesi. So. Do you think like developing a stem cell based product is more difficult than developing a T-based cell therapy? Is there any kind of correlation with like cell source, cell type, even therapeutic indication from a regulatory point of view?
It's a good question, Fabio, and I think the answer is that one type of product is not necessarily more difficult to develop than another. Maybe if I was to explain why certain products may be more difficult to develop than others, it would be in the case of genetically modified cell therapies compared to cell therapies which are not genetically modified. And that's simply because you need to also modify the cells as well as expanding the cells too. So usually for genetically modified cells, they're modified they're engineered with viral vectors. There are now non-viral vector approaches that are under development as well and becoming more prominent. But there's a mechanism for genetically modifying cells as well. And with a vector, then you have to manufacture a vector as well. And the amount of development work, CMC development work that's involved in developing a vector is really the same as developing the cell therapy itself. So, in many ways you're developing two products in one. But I think I wouldn't necessarily try and argue that a stem cell therapy is more difficult to develop than a T-cell therapy or vice versa, because each product has its own product-specific challenges that need to be understood. All cell-based therapies begin with a starting material from which cells are enriched, and then those cells are usually expanded in culture. They may be genetically modified, etc. The important thing about any cell-based therapy is to understand what product you want at the end. So starting with a mixed population of cells in the starting material, in the tissue of interest, you need to get a particular cell population at the end that mediates the pharmacological activity. So you need to understand how to design a process to go from starting material to the finished product, but then understand as well how the process is impacting the cell population that you're working with. Impurities that are there in the starting material at the beginning, how they may persist in the product even if you're enriching for a particular cell type, how process changes may impact those different populations of cells in the starting material. So it's really the hard work is about characterizing each particular product and designing a process that will deliver the right cell type that you want with the desired pharmacological activity.
Thank you, Anthony. I learn a lot, and I'm certainly very pleased to see where we are now as an industry compared to where we were like 8 to 10 years when let's see, we've seen the first CAR-T product approved and the first kind of stem cell based product approved. So thank you so much for your insights today, and I learned a lot, but I'm looking forward to learning even more from our workshop. And until then, bye bye.
Fantastic. It was great to speak to you. Thank you, Fabio.
Thanks.